First of all, I thank the organizers, especially Laura and Turan. They gave me a great support to get all things here ready before you guys arrived. And it's a great, great pleasure to be here. I think that commonly we spend quite a lot of time within our groups and talking with our peers, which is very important for science to develop. But it's also very important that we have this dialogue with people from different areas, so we can always get again and again a feedback and a different perspective from what we've been developing so far. And I think the opportunity of being here in the Scientific Café, what has been created now, is an excellent opportunity for that. I'm also very excited to be here in this historical building that I've seen only from outside, and it's a beautiful place to hold such a high quality meetings. Today I'm going to talk about functional and dysfunctional brain networks, and I work at the Systems Neuroscience and Imaging Psychiatry, SNP we call, lab, that is at the Department of Psychiatry and Psychotherapy of the University, so the Medical University Göttingen. So we have here the description of our working group. And I wish I could just show you the link there. All the information of all the projects that we develop is in there in more details. I'm not going to talk about all these 15 projects we're running at the moment, but all this information can be also found in English. And I think it's for anyone who's wished to have a better look at that, please go to it. I have always been puzzled, been intrigued by how complex then can the brain be. I think this is a topic that always fascinated me from the beginning, and that's probably what motivated me to be working in this area. I have a formation of a psychiatrist, but I always wanted to combine those two things. So I think that when we better understand how is the organization and function of the brain, we probably can develop better in the medical side as well. And so I'll just present here some questions that I wish to discuss today in this talk, and I will try to more or less answer them, but they are not definitive answers. They're not complete answers, because we are now talking about the edge of the knowledge of what is being developed so far, and I'm interested in hearing what the audience has to tell afterwards. So first one is how to understand the brain system's organization and function. The second one is, do brain networks communicate and dynamically interact? Third one, what are dysfunctional networks? Is fully decoding the brain system a precondition for an effective intervention? And lastly, is personalized intervention an important aspect of a successful therapeutic response? I'll start with the first one, and I will go back to the beginning of 1900, when very important works from Brodmann were published, trying to separate, segregate, understand which regions belong together and where are basically the limitations between those regions. And this was done histologically by a very systematic, incredible work separating the cytoarchitectonic distribution of assembly of cells, and that had this commonality in features, and they would just correspond to these different numbers that stay in there in the colors. Even nowadays, we're still struggling with this, and even though this being such an important work that even nowadays we use as a reference when we are actually studying and describing a specific region, people have been working also on the development of more and more particularities of those regions. So this is not a finished work yet. But then, how do we understand the brain other than just slicing post-mortem brains and trying to understand how the cells look like and how they are distributed? Another way of understanding this is also investigating what happens to people when they have certain traumas, for instance, car accidents or ischemias in particular areas of the brain, and which functions of the brain are then impaired. And this is a much recent work that collects a lot of patients together. And here you can see parts of the brain that have been affected by such events, and in the colors, which areas have been affected from cognitive control, decision-making, and so forth. And also, perhaps a very, the most innovative step has been functional magnetic resonance imaging, which has allowed us to have the brain mapped in vivo. So we don't need anymore only to slice the brain post-mortem, nor observe how certain traumas to the brain impair them, but we could also have the subjects inside of the machine, as you see here. And during a scan session, that can be repeated as many times as we wish, because this is only with a magnetic field, and there's no radiation or tracer or any substance that has to be injected. We can do different experiments with people inside of the scanner and observe how the brain reacts. It uses a very interesting mechanism, which is the activation of the cells and the need them to be perfused right after. So this mechanism is coupled, and the machine is sensitive to these changes of perfusion. So basically, these really, really tiny vessels that are there are being changed all the time when I'm actually activating different regions of my brain. And the perfusion that comes after is recorded by the machine. And a very simple experiment was done, for instance, with opening and closing eyes and seeing a visual stimuli. And we can see, for instance, that when we have eyes closed, they are definitely different in terms of activation, the direct activation, than we have them open, in a sense that we can actually track the regions of the brain that have been responsible for this activity during the experiment. So this technology can be today, in terms of spatial resolution, as small as having cubes of 1 millimeter or smaller to 3 millimeters, which is generally mostly used. And the resolution, the time that we scan the brain entirely for all these tiny bits of information, we scan it every 2 to 1 seconds, or even shorter time. So of course we can test people doing different things inside of the scanner, and here that's what we've been doing for long. In this study they show a very interesting overlap between different cognitive capacities, and interesting to see here is that they totally overlap. Generally, it has been thought from the beginning and theorized that a specific region would correspond to a specific function. And what we see then is that a specific region is actually involved in different functions according to the moment that actually has been performed and to which other areas it has been activated. So we go to more complex combinations of areas that basically perform different things. And this leads us to the next concept, which is functional networks. So nowadays we understand the brain as modular concept of different regions that are combined and preferentially work together, doing a number of certain functions. And as we see here, for instance, this spot, it rather work together with this and this and this, whereas this other one works together in combination with these ones. They preferentially work together. This means they are activated and deactivated a long time in a similar way. There are many networks that have been described so far, and each of those, as we see here, visual, auditory, and other more complex networks, they, sensory, motor, and other more complex ones, they assume they are responsible, they are specialized in doing different things. Okay, so do these brain networks communicate and dynamically interact, or they are just there, actually, each one of them doing their work? They do. Of course they communicate. And here is an interesting picture that shows, for instance, that shows this area in red and this other one in these warm colors, and they are plotted here as those warm colors. And we can see that a long time they are activated and deactivated many times. But another network that is here represented in blue shows exactly the opposite. So we can say here that it's an anti-correlation, and it can only happen if they basically know or they talk to each other. They can actually communicate in a certain sense. They have to be coordinated, that we basically end up seeing something like this. To give a little flair of how complex things can be and what we are actually digging into, I show this, but this is not actually the main idea today, to go too complex. But if we would, a matter of fact, just segregate any of those regions, all of those regions actually, and the brain with different colors and list them in the X and Y here, same axis, we could also evaluate how much each of those regions correlate to another. So when we see a high correlation, this means they are actually activated and deactivated similarly over time. They are shown here in hot colors. On the other hand, as we've just seen the previous example, when it's the opposite, the anti-correlated networks, they're seen here in cold colors. And we have kind of a picture of the whole brain in a certain moment. So let's say this was done in 5 seconds during the scanning time. The interesting part is, and here you see how complex that is, a long time this changes. So this picture of the brain along this 5 to 6 seconds is clearly not the same afterwards when time passes. And actually, this is a very interesting study. They try to break these networks in time and see how these states, these micro states as they call, can be segregated from one another. And it's actually that we have 6 minutes, indeed, of scanning time and smaller than 1 minute time is probably not so much difference to be seen. So this is how dynamic they go a long time. And it's clear that our brains challenge day to day, every time, to different things. And it's clear that basically this dynamic should exist. That we, of course, give the better and specialized networks to a given challenge and then swap them whenever this is not necessary anymore. So what are dysfunctional networks? The functional networks, for instance, are displayed here, and we call them the sense because they allow us many things, including learning capabilities. This is a nice study of a group of people that have been challenged to learn for an exam. And once they did so along months, their networks have been tracked and they are specialized. It's clear that once you try something and you repeat it and again, things are not as difficult as the beginning. And also we develop certain skills, let's say when we're studying for an exam. And after a couple of months, we actually see the difference here of regions that actually are much more connected than others. Good. But back to dysfunctional, what is then that? This is what we see generally when we're talking about psychiatric patients that are not only having many symptoms that bring them to us, but we also see a lot of impairments. Dysfunctional networks, for instance, as we see here in this study, they are signalized, they are in blue and in red, and they show differences from the controls that we're seeing in patients with depression. And another study, for instance, showed that these patients also have learning impairments. So if we give them a motor task to perform and we evaluate how much they can learn from this motor task overnight, which is actually what was done here, we see that in green, controls can learn this much more when they repeat this task, but patients with depression, they learn much less. And perhaps this has to do with this limitations that they had, this dysfunctional networks that were in the background. This is a study we did tracking the activity from the hippocampus, which is a region very important for learning and memory, and it shows this impairment shows a clear correlation to the connection of those prefrontal areas. Do we need to fully decode the brain? Do we need to understand exactly how it works so that we can after intervene? And the answer is no. We've been doing so with medication, for instance, but also with other types of approaches, and we've been having good results. We don't even know if we will understand the entire functioning of the brain in my lifetime or your lifetime. So basically, we have to give everything we have to do so, but we can also, in parallel, develop what is possible to be developed for people who are suffering from a certain disease. And that's what has been done so far. So TMS, transcranial magnetic stimulation, for instance, is a coil that is electrified, and it creates a change of magnetic field as being displaced in there, and very locally create changes, so makes little electric stimulations to the regions through the skull. So basically it's known invasive in this sense, and it's been used and approved for patients with severe depression that do not respond so well anymore to any kind of medications we've been trying. And from some of those patients we had a really good result. From other patients, not so much. And it leads to the next point, which is, are we performing TMS in depression in the best way we can? Nowadays, we identify the region that is responsible for the motor activity, and roughly 5 centimeters in front, where a region called dorsolateral prefrontal cortex is, we stimulate. But of course, the variation we see, for instance, in each one's face, this means we all have faces, we all have eyes, noses, mouth, but they vary in terms of spatial distribution. This is also to be expected in the brain. And it perhaps is too imprecise to consider only this 5 centimeters in front to be stimulating. It would be much more precise if we could map someone's brain and stimulate the specific region that we believe is actually not working so well. And this is what is being suggested by these authors in here. And this is the moment, at the moment, the actual situation. We believe it has to do with the networks in the background. And of course, that personalized intervention, so tailored to each person, to the anatomy, to the functional distribution of the networks in someone's brain, but also so all the impairments that have been seen in the background should probably lead to a more successful therapeutic response. And I'll just bring here the last point, which is how much individual variability can represent. So we have all those networks again, here mapped in a group. In contrast, we have those networks seen in an individual, and we see how much fine-grained differences we see in here, so much more details in comparison to the top ones. So if we actually go for somebody's brain, is as a matter of fact, very similar to the group brain, but it's not exactly the same. And if we go for more personalized interventions, as suggested in here, we could first map someone's brain, then we stimulate a specific area, spatially and also functionally, that needs to be stimulated, and we also track the differences that we've been seeing on this. This will be a reasonable, structured way of using TMS, and that's what I expect to be performing very soon. It's a pleasure to have you here today, and thanks a lot for your attention. I hope we can go to also interesting discussions about this.